Wow, is this an exciting meeting or what? I mean, I've never heard anything like that gel talk. I'm a nutritionist, and I think everyone in this room is a nutritionist. How many of you eat? Yeah? Okay, so you're experts. The trouble with vitamin E is it was discovered almost 100 years ago, and there's controversy. What does it do? Why do we need it? So that's what I'm going to talk about. Why do we need vitamin E? What does it actually do in the body? How much do we need? And oh, by the way, if you saw those reports that vitamin E will kill you, I'm here to dispel that rumor. It's not true at all. We've been trying for about 5 years to kill rats with vitamin E, and it can't succeed. But that comes later. We're going to start off with why do we need vitamin E? And lipid peroxidation. That is the most dangerous kind of free radical chemistry. This diagram that I'm showing you was actually developed by my colleague Graham Burton, and we published this over 25 years ago. And it's really quite obvious that you have an initiating event and make a carbon-centered radical. Well, I'm a nutritionist. I said to my chemist friend, what do you mean, initiating event? And he said, well, you know, like gamma rays from outer space. And I'm thinking, well, how important could that be? Well, it turns out the mitochondria are converting food energy into energy the body can use. And they do that through the electron transport system. That's how the body is generating carbon-centered radicals. The problem is, in the presence of oxygen, they make a peroxyl radical. That radical abstracts hydrogen from polyunsaturated fat. When that happens, you end up with a lipid hydroperoxide and another carbon-centered radical. When you get that second radical, now we've got a chain reaction going. Lipid peroxidation is dangerous because it is a chain reaction. And the only reason you don't turn into a pile of rancid fat is because vitamin E, our hero, comes in and reacts with that peroxyl radical 1,000 times faster than it can react with polyunsaturated fat. Well, that's been known for a long time by the chemists. Food chemists know this really very well. So you need vitamin E to prevent you from going rancid. That's the bottom line. It's kind of disappointing for all those molecular biologists, but I'm sticking by that. Now, what we did is we took zebrafish. Why? Well, when vitamin E was discovered, they started with E-deficient rats, made them pregnant, and those pregnant rats couldn't carry the fetus to term. So does the rat need the vitamin E, or is it the fetus that needs the vitamin E? Well, if you have a fish, the fish is only going to make little eggs that get fertilized, and they're outside and separate. We can study the embryo. So we made a vitamin E deficient diet, fed those fish, and we got deficient embryos. And what happened? Well, very bad news. 50% of the embryos died by 5 days. By 5 days, you ought to have a swimming around zebrafish. And you can see on that slide how ugly the deformed fish were. So what we learned from our zebrafish studies is that vitamin E is critical. Here you can see the vitamin E content of the embryos. When they are first fertilized, the E-deficient ones have about 40 times less vitamin E, and over those 72 hours, the vitamin E is decreasing. If we look at DHA, that polyunsaturated fish oil, these fish we are actually make their own, and you can see the mother is really trying very hard. Those black dots are the E deficient ones. The E deficient eggs actually have more DHA, but they decrease over time, and that's why we think the zebrafish gets into trouble. So, polyunsaturated fatty acids are protected in vivo by vitamin E. Now, the other thing we discovered about the zebrafish, which is very interesting, is that the tocopherol transfer protein. In humans, it's only in the liver, and so far it's been detected in the brain. But here we're showing you that the message, so messenger RNA for this protein, is in the brain and eyes, and the black and white picture shows you that vitamin E itself is localized in the brain and eyes of the zebrafish. So what happens if you knock it down? Then you should have no vitamin E in the zebrafish embryo, even though the egg has lots of it. So the egg yolk has lots of vitamin E, but lacking this protein, it can't transfer from the yolk into the developing animal. And what happens? Bad news. You end up with a zebrafish where the brain doesn't develop, the eye doesn't develop. And this happens at about 12 to 15 hours. I wasn't brave enough to show movies to you guys, but if you want to see the movies of this, go to PLOS ONE. It's online. You can see the developing animal. What's important about those 12 to 15 hours? It turns out it's the same time as the rats lose their babies. It's also about 20 days in a human pregnancy. So before a woman knows she's pregnant, she needs vitamin E for that developing fetus. So we can conclude that vitamin E is protecting the brain. How much do we need to do that? Now it's getting harder. The Food and Nutrition Board in the United States came up with the idea that if we use the data from studies done in the 1950s by Max Horwit, we can come up with a number of 12 milligrams. You add 2 standard deviations, that gives you 15 milligrams, which is kind of roughly equal to about 30 IUs of synthetic vitamin E in your vitamin E pill. 1,000 milligrams is too much, based on very iffy data. Who gets that much? Well, this is the problem. 96% of women, 90% of men do not get even 12 milligrams a day. So we have a problem. Either the RDA is too high, so we don't need that much, or there's more food than we think, or everything's wrong and people need more vitamin E than they're getting. So how do we tell? I looked at the literature. This is a very complicated picture because it's not a standard picture. The x-axis has how many articles I read, and the y-axis is plasma alpha tocopherol. And there are roughly 100 articles out there reporting deficient levels of vitamin E in the plasma of people. And most of these people are children. So I think that's very concernful. We have the problem that someplace between 20 and 30 is considered adequate amounts of vitamin E in the plasma. But what about those people between 12 and 20? That seems kind of iffy. So we need a better marker. And so one of the things we've proposed is to look for a new biomarker of vitamin E, and that is to look in the urine. Turns out that once you have a little too much vitamin E in the liver, it starts metabolizing the vitamin E, and we excrete it in the urine. So if we collect the urine, we can tell is somebody adequate or not, hypothetically. So this is the pathway for all of you who haven't heard about CEHC. It is carboxyethyl hydroxychrominol. We're going to call it CEHC. You start off with your vitamin E molecule, you hydroxylate the tail, you go through a series of beta oxidations to get to this molecule. In order to use it as a biomarker, you need to know three things. What is the diet, what is the plasma, and what is the urine? And it would also be nice to know some health benefit, but one step at a time. So I collaborated with Lenore Arab at UCLA, and she had this wonderful study. She trained people in Los Angeles to do diet histories. Oh my God, I will not fill out one of these things. These people did 8 of them, and they are to report what did you eat in the last 24 hours. And not only do you have to know what did you eat, how much did you eat? And so you have to report exactly. And they used this computer program, and they did an elaborate job, and golly gee whiz, yay, we ended up with people in Los Angeles can actually eat the RDA worth of vitamin E. But if we look closely at those numbers, oh dear. 15 milligrams, yes, on average it looks good, but it's plus or minus 13. Not so good. The plasma was 25 plus or minus 8, and my biomarker, 2.9 plus or minus 3.2. Altogether, a big mess. And if you look at the data, you can see these are the people who are not taking supplements. These are only people eating the diet vitamin E. And if we look closely at this critical area down near the bottom, we want to know, does this biomarker change as the amount of vitamin E consumed increases? And our answer is yes, that about 1.4 micromoles of CEHC per gram of creatinine is an adequate indicator of alpha tocopherol metabolism. That goes along with an intake of about 12 milligrams, 12.8 milligrams. So this looks good. Only problem with it is we don't really know how much were these people eating. This is a hypothetical diagram of what we think ought to be going on. If you're eating around 10, you have plasma vitamin E on this left y-axis of about 12. Eating 400 IU pill of vitamin E will double to triple your plasma concentration. Everybody agrees that's true. If we look at my biomarker, very small changes at low intakes. As you start increasing, and this is why vitamin E is not dangerous, it gets metabolized and excreted in the urine. It's not being accumulated in the liver. Now, the fact that these two lines cross doesn't mean anything. It's just to show you that while the plasma is plateauing, the biomarker is being excreted. So we can actually use this, we think, as a good measure of adequacy. So we have ongoing studies. We're trying to do this in a very carefully controlled way with deuterium-labeled vitamin E, where we can measure the metabolite, measure what's going on in the plasma, and know exactly how much do people need. I'm thinking that RDA is right. All of our data still are supporting that 15 milligrams a day. So if you're one of those people who doesn't like eating nuts and seeds, doesn't eat a lot of oil, so olive oil is not your choice, maybe you ought to be taking a vitamin E pill. And that's where we get to the question of how much is too much. Now, although I am not a fan of all those meta-analyses that say vitamin E will kill you, there are some carefully controlled clinical trials with vitamin E that have shown bleeding, hemorrhagic stroke, heart failure, and I guess the latest excitement is prostate cancer. How do all of those things, and different trials have different results. How can that be? And my answer is, it's all related to vitamin K. So we have to digress and learn a little bit about vitamin K for a minute. Vitamin K is involved in carboxylation of vitamin K-dependent proteins. These proteins have very specific glutamic acids that get gamma carboxylated, and they're called GLAs. And those GLAs are involved in calcium chelation. So for all of you chemistry fans, this is a 4-electron oxidation to make a K epoxide, and that epoxide has to be reduced back to the form of the reduced K. So the form in your diet is kind of the intermediate phyloquinone. And all of these vitamin K-dependent proteins require this reaction, and they require multiple of those GLAs to be made. There are 17 known proteins. They're involved in blood, in bone, in apoptosis. That brings us to cancer. There are a bunch of them that people don't even know what they do yet. So there's real possibility. So why do I think vitamin E has anything to do with vitamin K? Well, it's been very well known, probably more than 50 years, that if you feed a rat high vitamin K diet, or excuse me, high vitamin E diet, then that rat can have bleeding. We've measured tissue levels. The tissue levels of vitamin K are actually down. We looked very hard to see, is E causing K to be metabolized? Is it causing it to be excreted? What's going on? Could it be interfering with that recycling process? None of those things were true. How does E decrease K? I'm sorry to tell you, I don't know. We've got some exciting ideas. We're pursuing those actively. In fact, I got preliminary data yesterday I'm very excited about. I'm not going to tell you about it, but you can invite me back in 2 or 3 years, and we'll tell you the answer for vitamin K. Meanwhile, this sculpture is the Linus Pauling Institute's answer to Stonehenge. Once a year, the light is exactly right, coming in through the window to light it up, and it makes rainbows. The rest of the year is kind of bland and boring, so I thought I'd show you that. So, what do we know about vitamin E in human health? I think probably the RDA is right. 12 to 15 milligrams is correct. The big problem is most people are not eating nuts and grains and seeds, and they're not eating green leafy vegetables. Probably the biggest problem with that recent SELECT trial where vitamin E caused prostate cancer is they picked a bunch of men who eat meat and potatoes and not their green leafy vegetables. So start eating salad, gentlemen. I showed you that vitamin E is incredibly important for the brain. As we have the aging population, what we're finding with the zebrafish is there are specific phospholipids that contain DHA that are actually decreasing. And we need to look further to identify exactly what they are, but that's on track also. Now, Alzheimer's risk. We think vitamin E is important to protect you against that. And finally, this is an important point. We took wheat, milled away the wheat germ, discovered people got niacin, thiamine, riboflavin deficient. Then a while later, we discovered, well, there's not enough folic acid. So let's put those things that we took out of a whole grain back into flour. Well, if you remember, wheat germ oil was one of the original sources for vitamin E. And I would really like to see food fortified with vitamin E. So this is my current lab group. From the left, Jae Woo Choi is in charge of our oxidative nitrosative stress group. Catherine, wearing her beaver shirt. Beavers are the name of our team. She and Melissa over here in red are the two new nutrition graduate students. Catherine's working on vitamin K. Melissa's working on the zebrafish. Scott Leonard has been working with me since the first day I walked into the institute, and he's absolutely essential, and he's helping Emily learn everything there is to know about nutrition. We've had a bunch of great students and collaborators. Our work is supported by the NIH, the USDA, and now DSM, so I thank you very much. If you'd like to learn about micronutrients, the Linus Pauling Institute has a website that has all kinds of information. Please go and visit our website and learn about micronutrients. With that, I'm going to stop and look forward to your questions. Thank you very much.
Thank you very much, Professor Maret Traber. I think the key thing I heard you say was to go nuts about nuts.
Exactly.
Go nuts about nuts, ladies and gentlemen. Your questions for Professor Traber. Who will be first? Yes.
Thanks for sharing this presentation with us. I was intrigued by your hypothesis that vitamin E deficiency could be linked to Alzheimer's diseases. Could you elaborate on that a little bit?
So we think a lifetime of less than adequate intake of vitamin E is leading to a lack of protection in the brain. It turns out that the tocopherol transfer protein that I said is expressed in the liver is also expressed in the brain. It's involved in keeping the vitamin E next to the Purkinje cells. And in E deficiency, you actually see a dying back of the peripheral nerves up through the spinal cord into the brain. Now, in general, if you try and put an animal on an E-deficient diet, the E is retained by the brain probably because of the tocopherol transfer protein. But if you don't have any E getting into the brain and accumulating radical damage over time, this is where I think that we have the real possibility that it's an inadequate intake, a lifetime inadequate intake of vitamin E. So at the moment, it's speculation. If you look at the recent data on the biomarkers predicting Alzheimer's disease, some of those biomarkers are in fact phospholipids with DHA in them. And a lower level of those is predicting the incidence, the risk of Alzheimer's disease. There's another study that was done in the Framingham study, where it was the opposite relationship. High levels of DHA with phosphatidylcholine with DHA was associated with a lower risk. So this balance of having a phospholipid with DHA, so DHA is the other solution to this problem, eat more fish oil, and protect the brain.
Yes, I would like to remind the online community that you will have a chance for a Q&A online later with Professor Traber. Yes, Herman.
Yeah, thank you for your very clear presentation. Maybe I have a bit of a fool's question because I'm not a nutritionist. I was fascinated by the fish experiments. So I wondered if there is a relationship between the deficiency of vitamin E and premature birth, or even getting to a healthy pregnancy.
That's kind of the implication I was trying to make, that in fact, one of the concerns about 96% of women not eating adequate amounts of vitamin E could actually lead to difficulty having, essentially you end up with your miscarriage before you even knew you were pregnant. I think it's possible. There's work in the developing world that may in fact be true. I've recently read a manuscript that I said, please publish this as soon as possible, because I think there's data to really be concerned that we look around the room and say, well, we're all fine, so we don't really need more vitamin E. It must be that the RDA is wrong. And I'm out there clanging the bell saying the RDA is right. We have a problem. We can solve it by a pill that costs 10 cents a day. Please pay attention. And the medical community is going the other way. It doesn't cure heart disease, so therefore it couldn't be important.
Yes. People on the steps, be a bit more active. That would be nice. Yes, sir.
Professor, I was wondering about your statement you were making, or the data you were suggesting that indicates if you have a diet enriched in vitamin E, you have a low amount of vitamin K in the tissue. The data is coming close to the 50 milligrams intake per day of vitamin E. What's a higher amount of vitamin E that will lead to this deficiency in vitamin K? And in that way, then what would be an adequate fortification amount that you would suggest?
So those are excellent questions. They're all numbers, and these numbers are all hocus pocus numbers. So having said that to begin with, in order to develop vitamin K in a rat, K deficiency in a rat very quickly, we were injecting huge amounts of vitamin E. However, if we take a look at the Women's Health Study, this study gave 40,000 women 600 IU of vitamin E every other day for 10 years. They pointed out a couple of benefits. 10% of their women were over 45 to start with. They actually had less heart attack risk, but everybody overlooks that because they say, well, we already know vitamin E doesn't help with heart attacks. The other part that was very exciting was there was a decrease in thrombotic risk. So over the 40,000 women, this is a placebo-controlled matched trial, used vitamin E versus aspirin versus placebo. And what you saw was the women who were taking aspirin had a decreased risk, I believe it was around 26%, decreased risk in venous thromboembolism. So these are blood clots in the veins. And they went on in the discussion of that paper to say vitamin E is safer than warfarin as an anticoagulant. So nobody is measuring K in those studies because K levels in the plasma are 10,000 times lower than vitamin E. We've had to really develop mass spec methods that we can even play in the vitamin K game so far. And our thinking is that it's because K is so well recycled, you don't need very high levels of it. There is no RDA for vitamin K, and so we're up in arms about how much do you need. So I think the easy answer is actually vitamin K is very safe also. And it's very safe because its job is to gamma carboxylate those proteins. The proteins are regulated to do what they're doing. So in that case, you can't get too much K, and it looks to me if you put an E and a K in a pill together, you've got the problem solved. But I just have to prove that other part that that's what the problem is. So that's why I'm saying give me a couple more years and then invite me back.
Yes. Anybody on the steps? Raise your hands if you have a question for Professor Traber. This is really your moment. She'll be going back to Oregon State University in a matter of days. So.
Wow, I hardly just got here and it's so
well,
beautiful and I'm having
so much fun. You're welcome to stay as long as you wish, really. I'm just trying to stimulate them. There's a question for you there.
Okay. Yeah. You're looking into supplementation for the vitamin E, but is your marker, your biomarker, looking for sufficiency of vitamin E in the human body, something we can actually roll out and have in our routine blood checks?
Okay, my biomarker is in the plasma, but it's really the place to look for it is in the urine. It turns out that it's hard to measure because the body really likes alpha tocopherol and makes very small quantities of the stuff. This is micromoles per gram of creatinine. So it's small concentrations. What we're trying to do is give the Food and Nutrition Board, the Institute of Medicine, data that they can set the new RDA and be able to say that this is the amount of vitamin E that you need, and then we can quit with having to measure those other biomarkers. It may be interesting for research, but as a practical point, it's really kind of hard to measure that. And so it's a, how do we get data? So the next RDA is really based on science, not studies that were done in the 1950s. And that's a whole nother story.
Anybody this side, maybe? Yes. Excuse me while I nimbly walk through this small corridor.
Yeah, I was wondering about that transport protein, which is that all over expressed, or is that a recruiter for vitamin E, so you can use it as an indicator for tissue in need?
So the alpha tocopherol transfer protein is largely only expressed in the liver. There have been a relatively few studies looking at the brain, but it's largely the liver. And if you knock the protein down, or if you're looking at humans that have a genetic defect in that protein, and that was how it was discovered, those people have vitamin E deficiency. What the protein does is you eat your bowl of almonds, the vitamin E gets transported to the liver in the chylomicrons. Once it gets to the liver, if you have that protein, it grabs alpha tocopherol and puts it back in the plasma. Turns out that if you're lacking the protein or it's defective, vitamin E just gets metabolized and excreted. And so you absorb it, you get it to the liver, which means the people who have a defect in the protein, you can give them vitamin E supplements, about 1 gram a day, and then they're absolutely fine. They have normal tissue levels, everything works fine. So the role of the protein is to salvage vitamin E and to protect it from getting excreted, which makes it also, both K and E, because of this recycling, makes it tough to establish how much do you actually need.
Thank you. One question here.
You are a supporter of enrichment of vitamin E, food with vitamin E. Similar like the folic acid case, and especially in the US, you have shown that it really works. The problem is in Europe, authorities are not very enthusiastic about enrichment of food. What is your second best option then?
Well, my second best option is a multivitamin pill, because it has both E and K in it, along with all of those other things that you're not so sure that you took. So that's the second best option. Of course, you could always do what your mother told you to do. Eat lots of fruits and vegetables, eat whole grains, eat fish, eat a nice, expensive diet, and then you can afford all this. But as we heard at the first lecture this morning, the whole world is expanding. We are getting to the place that maybe we can't afford all these fancy foods for everyone, but we want everyone to have optimal health. There was a very nice study from China that showed that they measured the plasma alpha tocopherol in babies at birth, and then at 2 years old, the cognitive function. The cognitive function of those children at 2 years old was higher with higher birth plasma alpha tocopherol. So I think this is really important for the coming generations, that we provide sufficient vitamin E and sufficient DHA.
Allow me a layman's question in between. Is there a reason why there is no K and E pill yet?
People think vitamin K is involved in blood coagulation, and blood clots are what cause heart attacks and strokes. You want a blood thinner, and in fact, warfarin is a drug that inhibits blood clotting. It turns out that people who are on warfarin are told, don't eat green leafy vegetables because your blood will clot. Well, the way they ought to be telling people is eat lots of green leafy vegetables. We'll adjust the dose of warfarin so your blood doesn't clot so easily. It turns out that there's a protein on the artery wall called matrix GLA protein. This protein chelates calcium. Little claws hold on to the calcium, and it prevents the calcium from precipitating on the artery wall. If you don't have enough vitamin K, that protein doesn't work. The whole artery wall calcifies, it hardens. We increase blood pressure. Oh my God, now we have heart failure. Good question.
Thank you, thank you. Thank you. Well, I try, yes. Time for one more, ladies and gentlemen. One more question for Professor Traber. I'm sure there's somebody here who really has a very pressing question for Professor Traber. Yes. Of course, it's the furthest from where I was standing.
Well, this way you get exercise.
Well,
this
yes,
is good
I am.
too.
Yes. There you go. Oh, sorry, yes.
Does winning the DSM prize affect your research in any way?
It makes me very pleased and proud. I do vitamin E research because I think it's fascinating. I started off working on vitamin E in the 80s because children were diagnosed with neurologic abnormalities due to vitamin E deficiency, and nobody knew why. And we started studying vitamin E using deuterium-labeled vitamin E that was provided by Keith Ingold and Graham Burton. And those studies led to the understanding of how vitamin E is transported, how does it get to all the tissues. And so I find it a fascinating subject. It's quite frustrating because people say, well, if it's an antioxidant, why can't you show us what it's really protecting? So that's my latest goal, to figure out what is it really protecting, and is too much really a danger? I think it was thrilling to get that prize. I never, ever imagined that anybody would really care enough to give me a prize. I just do this for fun. And it's quite exciting. You know, it's little puzzles. Why is this happening? And you make a guess. And yes, Marcel, I have to admit, I fell on my face a couple of times with this E and K interaction, and that's why I won't tell you what my latest idea is, because the last three have been completely wrong. And so maybe this one is going to work. But I think you were asking, you know, is the nutrition community now saying, well, your work is all supported by DSM, and so we don't believe you anymore? I don't think that's true. I think the funding situation is so hard now for biomedical science that everyone is looking for support wherever they can get it, and I think it's really important for science. And so this little tetrahedron that has the brain in the center, well, E is protecting your brain, and that is what I really think, and we can make everybody in the room smarter. Just take your vitamin E pills.
And go nuts about nuts. Thank you so very much.
Thank
Professor
you.
Maret Traber and Marcel Wibbolts would like to talk to you personally.
Okay, thank you. Thanks again, Maret, for sharing this passion that you clearly show on vitamin E with us. And I think, I mean, maybe we should use part of the corporate research program to fund pills for all of you guys. I think it would be interesting. So also for you, we have this token of our appreciation. We really appreciate you being here. We have a true, genuine and good winner of our prize. I'm convinced of that, and I think this audience as well. Thank you very much.
Thank you.